All right. So let's all focus in. So when we left off, we had begun our discussion of capillaries. We had finished up with our understanding of the principles behind how we vary resistance in the body, okay, which had been dependent upon our arterioles and how the smooth muscle surrounding them behaves. So I want us to do a quick review on those arterioles before we continue with our capillaries and try to understand this thing called net filtration pressure. Okay, so we're going to do a quick, not quick for points, just a quick jogging our memories, because you hadn't looked at this receptor stuff and this extrinsic regulation beforehand. You might need a little more practice before we get going. Yeah, and I don't care if what you guys put as your names in here, initials, whatever, it's fine. Okay, well, let's click there. Okay, so when we were talking about extrinsic regulation, one of the things we talked about was the nervous system. What part of the nervous system are we talking about there? All right, yeah. So we were talking about the sympathetic half of the autonomic nervous system. It's the one part that's regulating resistance as relates to those smooth muscles and arterioles. Okay. And we were talking about receptors related to this, specifically adrenergic receptors. Some of them are causing vasodilation, some of them are causing vasoconstriction. So which was the one that was doing the vasodilation? I'm just kind of walking you through the order I think is easiest to think about this concept of nervous system regulation of smooth muscle. All right. Okay. Actually, the beta 2 receptors. So if you want to make an arterial, bigger, you want to lower resistance, you want to increase flow, you want a beta 2 receptor in the arterioles leading to that organ. All right. So now that we know those cause vasodilation, where are we putting them? Where are we putting those beta 2 receptors? Okay, yeah. So these are the ones responding to even very low concentrations of epinephrine due to their high affinity for epinephrine. And we specifically want them in the arterioles going to your skeletal muscle so that you can do activities, your cardiac muscle, so that your heart can beat hard and fast, because we're activating our sympathetic nervous system. We're getting ready for our fight or flight response. Okay. So on the flip side, what are we going to want as a receptor if we're trying to constrict the smooth muscle? Okay. All right. Yeah. Beta 2 receptors for dilating, alpha receptors, particularly alpha 1 receptors for vasoconstriction. All right. So where are we going to look for those if we want to find a cell that only really has the alpha receptors and no beta 2 receptors on them? Okay, we got a little bit of a split here. So it's the rest of the body. So beta 2 receptors and some alpha receptors are found on the skeletal and cardiac muscle. We know we want lots of flow there as we start to activate our sympathetic nervous system. You don't really want to cut off blood flow to the brain, right? So that's why we don't see these alpha receptors in the arterioles going to the brain. We're not diverting extra blood to the brain during our sympathetic response, but we do want to keep our blood flow going to the brain. So it's really out in the rest of the body where we mainly see these alpha receptors. I don't quite want to say concentrated, but we're going to see those alpha receptors kind of spread throughout everywhere else. Okay. So what's our overall effect here? We're constricting out in the rest of the body, so lowering flow out to the rest of the body, dilating to the heart and the skeletal muscle. Okay, great. Yeah, and I'm walking you through like this. Hopefully, this seems relatively straightforward and simple because I know that the bullet points with all these different receptor types can sometimes get a little messy. But if you step back and think about what you want to do with your body under these conditions, hopefully it makes a little more sense, right? So we're diverting and increasing blood to our skeletal muscle and to our heart. We're constricting down everywhere else, so we don't have quite as much blood flow going there. And also we're able to keep our mean arterial pressure up. We're able to keep blood pressure up so that we don't like faint. Cool. All right, great job. Lots of 6 out of 6s. Okay. So that's where we ended off with the arterioles. That was one of our big picture topics at the end of last week. And we had introed our capillaries and some of their features. So we talked a little bit about the idea of exchange. We talked a little bit about how we can divert flow into specific capillary beds or avoid specific capillary beds by using meta-arterioles to bypass capillary beds, or shrinking down meta-arterioles and opening precapillary sphincters to kind of force blood into these little side routes. Okay. So hopefully people looked over some of this capillary stuff over the weekend. Okay. So we're going to be talking about net filtration pressures. So we want to make sure that we know what filtration is as we get started. Okay. So which of these things is filtration? Let's take a look. Okay, cool. Yes, it is movement from the blood into that space surrounding the capillary. So the first thing we're starting with are our directions, right? So when we have a capillary, overall, what we're going to be trying to figure out is how much do we have fluid moving out towards tissue into this interstitial space, right? And that direction is our filtration. versus how much do we have being absorbed, or sometimes we'll see it described as reabsorbed. For our purposes, the difference right now is not important. How much is coming towards the blood? So we're going to have that be our absorption direction. So today we'll be talking about a bunch of forces called our Starling forces. So we're going to be categorizing them all and thinking about each of them. Which direction are they pushing or pulling fluid, right? So that's what we're thinking about as we step back from the specific forces, because all we're going to try to figure out is on net, which direction is fluid going? Is it coming out of the capillary or is it coming into the capillary? Cool. All right, so we're talking about bulk flow across the capillary walls. So we're thinking about exchange specifically of fluid. This is going to be dependent on pressure gradients. It's going to be dependent on our forces for filtration. So movement from our capillary into the interstitial space, so that space between the capillary and the tissue, versus forces for absorption, forces moving us into the capillary. Okay? We're in general distributing our extracellular fluid this way. If you remember way back to like our first day of A&P, plasma, so that blood plasma was one of our types of extracellular fluid. This interstitial fluid in the interstitial space was our other type. Okay. A reminder of what those capillaries look like. All right, we've got a layer of endothelial cells. They're kind of glued together by something called a basement membrane. But really our big picture for capillaries is thin walls, just this one cell layer, easy to cross, right? And so we do want to remember, as we just think about all of these principles, what is and isn't able to cross capillary walls. Okay. So we can note small nonpolar things can always just diffuse directly across a cell wall. So we don't really have anything other than distance blocking things like oxygen and carbon dioxide, because they mix nice with our phospholipids making up our cell membrane. Okay. We do have pores in capillary walls, so spaces between the cells. So we are going to have ions able to passively diffuse in these spaces between when we've got pores, which in different capillaries may differ in size, and we'll talk about that in a little bit. And we can move proteins, but a protein is going to have to move by transcytosis. They're large. They're going to have to be kind of regulated, moving across these cell membranes through the process of transcytosis. Yeah. All right. So hopefully you looked at this ahead of time, did put it in your recommended reading. Okay. So we got filtration and we got absorption. Those are directions for filtration out of the capillary, absorption into the capillary. Okay. The next piece we're going to be thinking about is, what does a force for filtration look like? What does a force for absorption look like? And we're going to split into osmotic pressures and hydrostatic pressures. Okay, so let's see. We've got a couple of people who can tell us what an osmotic pressure is. We did talk about osmosis briefly in A&P 1, so this is part of where it comes back. Let's go ahead and take a look. Okay, yeah. So when we're talking about osmotic pressure, we're going to think about this for our purposes as a pull, right? So we're going to be thinking about cells or capillaries or spaces, right? But let's start with cells because that's how we thought about this when we thought about osmosis in general, right? So if you think about this, let's think about a cell with some ions distributed around it, right? So we've got some ions over here in our extracellular fluid. All right. Let's have some ions, just a couple of them inside our cell. These are going to be interspersed with our water molecules, right? So we're always going to have kind of an inverse relationship between the number of solutes and the amount of water. So here inside the cell where we have, in this example, relatively few solutes dissolved, we're going to have more water. Okay. So here are my waters. Okay. And outside the cell, where we have more solute, in this example, we're going to have less water. So you can think about this in two ways, right? Before, we might have been thinking about water moving down its gradient, right? So we had lots of water here inside the cell, less water outside the cell. So we could tell the direction that way, right? So we could see that we have water moving out of the cell in this example. But here, now that we're thinking about the blood, we're going to focus in. Notice that because of that inverse relationship, right, you can think about it as these solutes, these extra solutes, this extra protein or ion, depending on what we're talking about, it might be different, right? These higher concentrations of solutes pull the water towards where they are, right? This is how we can evenly distribute solutes in an aqueous solution, right? We can distribute them because we want kind of an equilibrium, an even ratio of solutes to water molecules inside and outside a cell when we talked about it before. Now we're going to be thinking about it with respect to capillaries, okay? But that osmotic pressure is that pull on water, okay? So that's going to be one of our two types of Starling forces, okay? I'll just write on the symbol for that real quick, right? Our osmotic pressures are going to be a pi symbol. And then as we move forward to fully expand our understanding of the Starling forces, we'll add a subscript telling you which osmotic pressure are we talking about, where are we talking about those concentrations of, in this case, impermeable solutes. So we'll now be thinking more about proteins because we know that the capillary walls can let some of those ions through the gaps. Okay, so osmotic pressure, a pull, right? A pull on water is one of the things we have to think about. And the other thing that we have to think about is the hydrostatic pressure. So this is going to be denoted for our net filtration pressure with a capital P, okay? You want to think about this hydrostatic pressure as a push, okay? So we're thinking we're filling up like a water balloon with a lot of fluid, right? It's pushing out on the walls, right? So our hydrostatic pressures are going to be pushes. Okay. So when we think about these, we're going to be thinking about each of them being in two places. We're going to be thinking about an osmotic pressure coming from inside the capillary, as well as an osmotic pressure coming from that interstitial space, coming from the fluid surrounding the capillary. And same thing for our hydrostatic pressure. We're going to be comparing the pressure of that fluid pushing out on the capillary walls, as well as the pressure of fluid between our tissues in that interstitial fluid, pushing the capillary walls kind of from the outside in. Okay? So in total, when we talk about these net filtration pressures, we're going to get to a total of 4. Okay, so 2 hydrostatic pressures, 2 osmotic pressures. And those are always going to be 1 for the capillary, 1 for the interstitial fluid. Okay. So in the case of our net filtration pressure, when we're thinking about osmotic pressure, we have to be thinking about things that can't cross our capillary walls, because if they can cross, they don't have to pull water, they'll just cross on their own. So we're going to be thinking about non-permeating solutes, which in the case of a capillary with that structure of pores is really going to be the proteins. Okay. So when people are emphasizing that they're talking about the pressure coming from proteins specifically, you'll see that denoted as oncotic pressure for our purposes, because the osmotic pressure for our movement across capillary walls, movement of fluid, is coming from the proteins. Those are going to be kind of equivalent terms, but that's why you see them switching back and forth. Osmotic pressure is the kind of general idea. Happens to be the case that when we're thinking about blood, it's specifically proteins. So we could also call it oncotic pressure. Okay. So let's think about those hydrostatic pressures first. Okay. That's kind of the newer one. So hydrostatic pressures, we're thinking pushing out on walls, or pushing into walls from the outside. Okay. So we're going to imagine our setup. Oops. Okay. So we're going to be thinking about our capillary. So we've got our blood. We're going to be thinking about our tissue over here. And we're going to label them because we're going to have to label these different pressures. Okay. So here's our capillary. And here's our interstitial fluid. Okay. Cool. All right. So when we think about blood coming in the capillary, we know it's coming from the heart into the arteries, through the arterioles, finally into the capillary. We know that there's a flow due to a difference in pressure between the arteries and the veins. Okay. But we got blood rushing in. It's slower than it is in other places, but there's still a pressure pushing it forward. And that blood in the capillary pushes out on the walls. Right? So we're pushing out on the wall of the capillary. Okay? So this means that here, back up, think about that direction for filtration and absorption. And you can always back up and draw it. I kind of recommend making sure that you know which direction is filtration and which direction is absorption before you start a problem. And if you did that, you'd notice that this out arrow is that filtration direction. Okay. So this is our hydrostatic pressure of the capillary. So P cap. And it's a force for filtration, right? So saying it's a force that favors filtration is just talking about that direction. So it's a direction towards the tissue, towards the interstitial fluid. Now it happens to be stronger at the arterial end than it is at the venous end. This is because we actually are, on the whole, going to be filtering some fluid out of the capillary and towards the tissue, right? So as we're losing some of this fluid moving towards the veins, we have a little bit less of a push as we get to the venual end. So you can see a drop from the arterial with a pressure of 38 to the venous end with that pressure of 16. But both of them are pointing in the same direction. They're both pointing towards our tissue because we're thinking exclusively about how much pressure do we have pushing out on the capillary walls. That's how we're defining this force. Okay. Now, still thinking about hydrostatic pressure, inside the capillary is not the only place there is fluid, right? So there is also some fluid, not a lot, but some fluid in the interstitial space, right? That's why we have interstitial fluid, okay? So here we have a hydrostatic pressure again. We're just going to specify that it's of the interstitial fluid. And it's still working as a push, right? So it's pushing on everything it's next to. So if we're thinking about directions for the capillary, that means it's pushing back towards the blood, okay? So this is a relatively low pressure under normal conditions, but it is a force that favors reabsorption. So it's that absorption direction moving into the blood, okay? So always think about hydrostatic pressures as a push, and then think about where we are pushing from. Okay. Now, questions about our hydrostatic pressures here before we talk about the osmotic pressures? Okay. we can see, just noted here, that we have a little bit of a gradient of the hydrostatic pressure here. So at the arterial end, if we were to add our total pressures together, kind of right at that spot, right at this spot on the arterial end, we would have a pressure gradient of 37, because we have about 1 millimeter of mercury as our pressure of this interstitial fluid. So all of these pressures are occurring kind of along the length of the capillary. We have a smaller gradient if we were to measure both of these forces over here at the venule end. Maybe I'll label that artery end, vein end, okay? Because we've had a little bit of a drop in that pressure, that hydrostatic pressure of the capillary. Okay? But in total, we've still got an overall kind of force for filtration. Okay. So now we're going to add on our osmotic pressures. Okay. So just like our hydrostatic pressures, we're going to have one coming from stuff in the capillary. We're going to have one coming from stuff in that interstitial space. But now we're thinking about osmotic pressure, so we're thinking about a pull, right? So we're thinking about solutes, specifically non-permeating solutes, pulling towards the location that that pressure is, okay? So we're going to have a osmotic pressure for our capillary, okay? So we're going to have a pi cap, okay? And in this case, what we're thinking about is that we have a bunch of proteins in the capillary, right? So we're going to be thinking about a bunch of proteins here in your blood. There are lots of blood proteins, okay? And they're solutes. They're non-permeating solutes. The only way they could get across the capillary wall under normal conditions is through transcytosis, which would kind of be regulated, okay? So we're thinking about them staying where they are, and they're exerting a pull on fluid, right? So here, we're going to imagine fluid being pulled towards those proteins, okay? So if we look at that direction of the arrow, it's into the capillary. The pull is pulling us in. So this is a force for reabsorption or absorption, right? That's our direction here, okay? Now, we don't usually have a lot of solutes in our interstitial space, but we do want to account for them in our total equation, because under pathological conditions, if you get an injury, right, some of this stuff might change. Some of it might not be exactly normal, yeah? But we're still going to be thinking about a pull, right? But now we're thinking we've got some kind of solute over here in our interstitial fluid. It exerts a pull on fluid, okay? So that is our osmotic pressure of the interstitial fluid. And looking at that arrow direction, right? We've got an arrow going from the blood into the interstitial space here. So that's a force for filtration, right? So if it exists, we've got a force for filtration, because that's just talking about the direction. Okay. So when we compare these, we're going to think about that capillary oncotic osmotic pressure not really changing. We're not going to be changing the number of proteins in the blood, right? They shouldn't be moving across the capillary wall. And we shouldn't really see changes to the oncotic osmotic pressure of that interstitial fluid either. We shouldn't have proteins moving around there much. So we're going to not have to think about a difference between the arterial end and the venous end here, right? We can just look at the comparison here. We've got overall force, 25 versus 0 to maybe up to 1 here. A force that overall favors reabsorption. So this pi cap, this capillary oncotic osmotic pressure, is rather large. Okay. Questions about this before I start erasing, actually? Cool. All right, hopefully this is straightforward. All right, so overall, we're thinking about forces for filtration and absorption or reabsorption. So we're looking at our directions, right? So if we look at our hydrostatic forces, we can see that the hydrostatic pressure of interstitial fluid pushing back into the capillary is a force for absorption. We can see that those proteins, that osmotic oncotic pressure of the capillary over here is a force for absorption. And in the middle here, we can see that our major forces for filtration are that hydrostatic pressure inside the capillary and any osmotic pressure we have coming from proteins dissolved in that interstitial fluid, right? So for these forces, we've got our absorption here in this image on either end. We got our filtration here in the middle. Okay. So what we're going to do when we put this all together to find our net filtration pressure, ultimately, we're just going to be comparing how big are our forces for filtration versus how big are our forces for absorption. This is just a straight comparison, right? So we're going to be adding them together, first at the arterial end, and then because we have a difference in hydrostatic pressure from the arterial end to the venous end, we'll think about what's going on at the vein end as well, okay? When we do that, we're ultimately going to see that most of the filtration is happening at the beginning of the capillary, so happening at that arteriolar end. And then we're going to have the forces for absorption kind of slowly take over at the venular end. Okay, so let's start by thinking about that arteriolar end. So I'll give you a minute to think about this. Okay, I'm going to tell you how to do this calculation. Okay. So what you want to do to get your net filtration pressure, or your NFP, okay, what we're going to be doing is we are going to take all of our forces for filtration. Okay, so you're going to add two numbers together here. And then you're going to subtract away any forces for absorption. Okay, so we're going to subtract away quantity. That's all our forces for absorption put together. So go ahead and start this by identifying, all right, what are our two forces for filtration? What are our two forces for absorption? So you're going to add those two together here and here. Then we're going to calculate across. I'll give you a little bit of time to do it yourself, and then we'll do it together. If you want to team up with your neighbor, that's okay. You can do that. You're doing that. I'm going to start drawing my little drawing in the bottom. If you're stuck on where to start, this might be what you do as well. And then you start by identifying directions for filtration and absorption, and then work your way through these quantities. Starting with those forces for filtration. So filtration is the out direction. So we're picking up pressure in the capillary pushing out. We're picking up osmotic pressure pulling us towards interstitial fluid. The other side, we're looking for our forces for absorption. So we see absorption is this down direction towards the capillary. And we can calculate across. Right? So we're going to put in all our numbers. For our total net filtration pressure here. We had 38 as our total forces for filtration, minus 26 as our total forces for absorption, giving us a total pressure for filtration, right? Because this number is bigger, of 12. Okay. Questions about this calculation? We'll keep moving a little bit. We'll keep this for our warm up for next time, doing the same thing at the venous end. Okay. Well, let's make sure that we have this all out in bullet points for ourselves. All right. So what you just saw was our calculation for net filtration pressure. So all we have to do, right, is identify our forces for filtration, that out direction, versus our forces for absorption, that in direction. and then add them all up. Add the filtration pressures together. That's going to be our first number. And we're going to add together our forces for absorption and subtract them away, giving us a total, right? So net is basically total pressure for filtration. We're going to see that at the arterial end, like we just calculated, we have a 12 millimeters of mercury force for filtration. So in total, our forces here on that first part are going to be bigger, right? So our forces for filtration are going to be larger, primarily because we've got this big force, this big P cap, this big hydrostatic pressure coming from the blood, coming into the capillary at that first part of the capillary at the arterial end. Okay? So we're going to start out with a force for filtration that pushes fluid, I'll say, into the interstitial space, so towards the tissue. Okay. All our numbers stay the same as we get to the venous end, but we do see a drop in that hydrostatic pressure, right? That's the one number that's going to change, the hydrostatic pressure of that fluid inside the capillary, because we lost some on the arterial end. So we're going to see a drop. And in fact, we're going to see it in total flip, right? So your net filtration pressure calculation, I'm going to point out, it's a pressure for filtration. You are calculating the force for filtration when you do this. That's why the filtration pressures come first. So whenever we get a negative number, and I'll practice a couple times in a little bit, or when I see you later, depending on how far we get in a second. But whenever you get a negative number, that means you don't have filtration, right? A positive number would be for filtration. A negative number tells you that that number you subtracted away is bigger, right? A negative number would mean your pressures for absorption were bigger than your pressures for filtration. So if you get a negative number on net, you don't have filtration, you have absorption happening. So in total, in reality, under normal conditions, across the total capillary, we're going to have more filtration than absorption happening, right? So it is true that by the time we get to the venule end, there's a little bit of a force for absorption in total, but it's not as big as that force for filtration was at the beginning of the capillary. So overall, you actually filter out about 3 liters per day of fluid out of your blood and into that interstitial space. Okay. So we, in this course, are not going to talk much about the lymphatic system, but it is super important because you don't want to lose 3 liters of fluid from your blood per day in total, right? That would not be good. You don't have all that much blood, right? So when we do mention the lymphatics, one of their big jobs, one of their big roles is going to make sure that you don't end up a big swollen mess with all your fluid in the interstitial space and like no blood left in your arteries and veins. So our lymphatic system, part of its job is going to be picking up that extra filtrate, so that fluid that we filtered into the interstitial space, we'll sometimes refer to as filtrate. So your lymphatic system can pick up that fluid, return it to circulation, put it back in your blood ultimately, so that you keep your blood pressure up, so that you have enough fluid moving through your vasculature and not causing you to swell up kind of everywhere. So just thinking about defining those forces again. Now, sometimes when we have injuries or when we have specific diseases, specific conditions, we will see changes to these pressures, right? That's why we're learning the whole equation, right? Despite the fact that some of these values we're going to see are usually about 0, it's not always going to be the case. Let's think about this situation. I'm going to tell you, if you get an injury, if you get a cut, right? You'll notice that you have a swell in the tissue after a while, right? You swell up sometimes, you get a bump, right? So part of that is because of increases in the hormone histamine, right? So this signaling molecule, basically. Okay. What happens when you get a cut or an injury? Your capillaries get kind of damaged, right? So your capillaries get damaged and histamine is responding and some more proteins are actually getting filtered into the interstitium, so into that interstitial space, into that interstitial fluid. Okay. So one of the Starling forces is going to change when we see those proteins in the interstitial fluid. So we want to define which of those Starling's forces is changing here. Logic. Okay, we got an array here. Okay, let's talk it through. Okay, so what's happening here? We got our capillary, right? And usually we have hydrostatic pressure of the capillary pushing out. We have any hydrostatic pressure in the interstitial fluid pushing in. We've got osmotic pressure in the capillary pulling towards proteins. So that's an in. We got osmotic pressure in the interstitial fluid pulling towards itself, which means that's a force for filtration out, right? So what we change here, one of the reasons you're swelling up is you're increasing forces for filtration, right? So what we have happen is you get more proteins, the histamine acting and this injury here in the interstitial fluid. Usually we're approximating that interstitial osmotic pressure is approximately 0. But now that we've got proteins here, that's not 0 anymore, it's going up. So now when we add together filtration pressures, we're going to get a bigger number than normal, which is why you see more fluid movement into those injured spaces. It's why you have swelling. Okay. So we will pick up here next time, practice a couple more calculations on this and think a little bit more about some conditions that change it before we move on to our venules and veins.